Hello everyone, and thanks for joining us today for the Cell and Gene Therapy 2025 Online Symposium. I'm George Hammond, and I'm here to moderate today's event alongside my colleagues Princess Sennett and Heather James. Before we start the talks today, I just have some general information and a few tips for getting the most out of the event. We would like to ask if you could please refrain from video, audio, or photo recording of all talks. Many of our speakers have kindly agreed to share their slides with you today, and these are available within the literature section on the platform. Please remember that we encourage debate and discussion and welcome all of you to use our networking services to introduce yourselves to other attendees. But we do not permit the overt solicitation of products or services and reserve the right to block any user who infringes on this rule. It's also incredibly important to say that this event was only made possible thanks to the generosity of our sponsors and partners, Acro Biosystems, BD Biosciences, and Sinobiological. For more detailed instructions on how to make the most of the features available to you at this event, please navigate to the event home and watch the overview video at the top of the page, or visit the FAQ section in the left-hand menu. And with that out of the way, I will now move on to introduce our opening panel discussion for the event. We're thrilled to introduce our opening session for the event, an exciting panel discussion on gene editing companies to watch in 2025. As the cell and gene therapy landscape continues to evolve, novel therapeutics are poised to redefine treatment paradigms and patient outcomes. This panel discussion featuring esteemed experts in the field will explore the most promising therapies highlighted in Clarivate's Gene Editing Companies to Watch and Drugs to Watch 2025 reports. It's an absolute pleasure to have you all join us today, so now, without further ado, I'd like to hand over the floor to Matthew to open the panel discussion.
Thanks, George. Good morning, good afternoon, and good evening, depending on your time zone, and thanks for joining us for today's panel presentation on cell and gene therapy in 2025. I'm Matthew Arnold, a recovering journalist and principal analyst at Clarivate, a company which uses data analytics and expertise to help life science companies innovate and answer patient unmet needs faster. So we are now 35 years out from the first successful application of a gene therapy, 13 years from the discovery of CRISPR-Cas9. The science of cell and gene therapy and gene editing are advancing quickly. So is investment in this space. In particular, we see a feeding frenzy around gene editing as large pharmas look to get in the game of this emerging field and not be left behind. The value of gene editing deals has increased steadily over the last 5 years, with the 5 disclosed blockbuster deals of 2023 approaching $25 billion combined, according to Cortellis Deals Intelligence data. Just recently, AstraZeneca announced a deal for an in vivo cell therapy company worth potentially almost $1 billion. So these areas are a relative bright spot in a very grim time for biotech and hold enormous hope for patients, particularly those with rare diseases and cancers. Our recent report from Clarivate, Leading Innovators in Gene Editing, which you can download at clarivate.com, identified 8 companies changing the drug discovery and development paradigm in this field. It found that IP creation is rising in tandem with financing in that space, with patent filings increasing 60-fold over the past 10 years, led by companies in the US and mainland China. Clinical trials for gene editing solutions have risen from 4 in 2019 to more than 30 last year, according to Cortellis Clinical Trials Intelligence data. Most of these were early phase trials, of course, with just a handful of late stage trials among the several dozen launched since 2019. So with us today are 2 of those innovator companies identified in the Clarivate report. First, we have Eric Kmiec, PhD. Eric is CEO of CorriXR, an oncology-focused biotherapeutics development company whose gene editing platform uses a unique CRISPR-Cas biomolecular tool to disable genes in tumor cells responsible for drug resistance. They're based in Newark, Delaware. Eric, tell us a little about yourself.
Well, thanks, Matt, and great to see Steve and Sophia as well. So I've been in the field of gene editing for about 3 decades, and I have seen kind of the good side, the light side, the dark side, and so over the past 10 years, I've been involved with a healthcare system in northern Delaware called ChristianaCare, which embraced novel genetic medicines for the development of treatment of oncology-based therapies, most focused on solid tumors. And so the Gene Editing Institute was created and it now has about 25 people in it, and we're essentially developing those kind of therapies a little further. About 2.5 years ago, ChristianaCare, which is the 14,000 person healthcare system in which GEI is embedded, decided to spin out their first company, which is CorriXR Therapeutics. And so we are actively engaged both in the development processing through the FDA, but also on the back end have a very good source for clinical trial development. So very happy to be here today and talk about this very exciting field.
Thanks so much, Eric. Also joining us, we have Steve Becker, Chief Commercial Officer of Cambridge, UK-based Broken String Biosciences, which is using its proprietary DNA break mapping platform to develop cell and gene therapies. Steve, like Eric, you've been around the life sciences industry for several decades. Tell us a little about yourself and your work and what you've
thanks
seen.
so much, Matthew. I appreciate the opportunity here to speak to the society today, as well as being on the panel with these great colleagues. So my background has been back and forth between the research tools and the diagnostic field. I've been with leading companies like Amersham back in the day when we were involved with sequencing the human genome, to most recently Thermo Fisher Scientific, where we were commercializing sequencing technologies and other technologies on a global basis. But I was brought into this field from my earlier work, like Eric had mentioned, that we've been at this for a while, and I think you mentioned it as well. But we saw that safety was a particular concern sort of in the last sort of revision of bringing these novel therapies to market. And that to me is compelling, right? As we continue to accelerate and bring these technologies to the field, we need a simple, elegant, and unbiased way to bring these technologies to market, such that the patients ultimately benefit and the society and payers know they're getting a great product. And so looking at the technology that we have here at Broken String, I feel that we're able to do this, and it just drew me to coming to this opportunity to really help accelerate the field and help patients get what they need.
Thanks, Steve. It's great to have you today. And also joining us is my Clarivate colleague, Sofia Ataide. Sofia is Senior Business Solution Consultant at Clarivate. Sofia, tell us a little about yourself and the work you do helping companies in spaces like cell and gene therapy and oncology tackle their business challenges.
Thanks, Matt. And hello to everyone joining this panel today. And hello, Steve and Eric as well. I'm really excited to contribute to the discussion today. So I'm Sofia Ataide. I'm a pharmacist by background. I have a master's degree in pharmaceutical sciences by the University of Lisbon and a master's degree in international health management from Imperial College Business School. I started my career at Clarivate, and currently I am a senior business solutions consultant for the life science and healthcare practice. And I engage with companies operating in the oncology and rare disease spaces by understanding their needs, their requirements, and long, short-term goals, and match those with Clarivate's wider solutions. So basically, my team and I, we combine deep domain expertise in the areas of clinical, R&D, and regulatory with technical knowledge of Clarivate's portfolio to support and generate the highest impact to our customers in many areas of their research. So it's a pleasure to meet you all, and I hope you enjoy this session.
Thanks, Sofia. So for our first topic, I wanted to talk a little about the latest breakthroughs and your insights into the clinical trial pipeline. But Sofia, can you tell us a little about what you're seeing in terms of approvals in the clinical trials pipeline in the space?
Sure. So the first thing I like to say is just to give some overview about some numbers. According to the American Cell and Gene Therapy, the therapeutic pipeline has sustained growth at approximately 6 to 7% in recent years. And this growth happens in the face of numerous development challenges, a lack of regulatory direction, as well as adoption uncertainty. So, however, we've witnessed significant milestones last year in the number of drug approvals and new clinical studies as well, as you've mentioned, Matt, in the beginning. To name just a few of these drug approvals, you have Ryoncil. So the first mesenchymal cell therapy received FDA approval for a pediatric graft versus host disease. It was approved earlier this year. You have Adaptimmune Stelcera, which is the first FDA engineered cell therapy for metastatic synovial sarcoma. And you also see other players in the field like Iovance and Autolus, who are also paving the way for more cellular therapies on the market. However, something that it's interesting to highlight is that not all new treatments have seeked the success that they were hoping for. And for instance, already happening this year, Pfizer has discontinued their hemophilia treatment, Becvez, due to the low uptake. So this was a major event in the way that Pfizer has emptied its entire gene therapy portfolio. So these are huge. The field has proven to be under huge momentum, and a lot of things are happening in the market space. But amid these developments, there's been a significant catalyst for innovation, which started with the approval of Casgevy in late 2023. So you all know that it was the first CRISPR-Cas9-based therapy to receive global approval. So this breakthrough has sparked a tremendous wave of interest, not only from gene editing firms, but also from big major pharma companies, all wanting a stake in the field, right? And also for these gene editing companies to advance their capabilities in this space. So Clarivate, as Matt mentioned, released last year a gene editing companies to watch report, which sheds light on the evolving landscape of gene editing and highlights 8 innovators that are disrupting the innovation, that are disrupting this field. But with regards to clinical trials, the gene editing therapies are seeing a surge in the number of clinical trials studied. So this growth has emerged from single digits just a few years ago to almost 30 trials starting last year. So basically, the number of trials that started last year nearly surpassed the overall number of clinical trials that have happened in the previous 5 years collectively, okay? And as well as Matt also mentioned, the majority of these trials are in early stages of clinical development, phase 1, phase 2, whereas we see phase 3 trials having started mostly to focus on confirming the outcomes of therapies like Casgevy. Okay. But something interesting to highlight that the report mentioned is that to note that fewer than 20 companies have initiated at least 2 clinical trial studies since 2019. And this means that only a small number of companies are actually driving the growth in gene editing trials, out of which about 1/3 have multiple assets in clinical stages. So some of these clinical trial pipelines that you see Intellia CRISPR gene therapy that received FDA clearance for pivotal phase 3 study for alpha-1 antitrypsin deficiency, and another programs in phase 3 for hereditary angioedema. You see CRISPR Therapeutics with several clinical programs for multiple lymphoma, multiple myeloma, and renal cell carcinomas being studied. BGene's CRISPR technology, which is developing a program to treat herpes virus keratitis, currently in phase 2 of clinical development in China, and as well Beam Therapeutics developing a base editing gene therapy, currently in phase 2 development for sickle cell disease. Okay, so these are just a few of the most breakthroughs companies in terms of their clinical trial pipeline. However, the ability for companies to also address key medical and scientific challenges, demonstrate proof of concept, collaborate with notable scientific academic institutions, maintain strong intellectual property, and also secure funding, also expands the coverage for more innovative companies to disrupt this field. And in the gene editing companies to watch report that Clarivate launched last year, these factors were used and analyzed to identify these 8 disruptors in the field, out of which 2 are present in this panel today: Broken String Biosciences and CorriXR Therapeutics.
So it's still a really an emerging field, a very small field, yeah. And we're fortunate to have you, Eric and Steve. You two are in, as our British colleagues would say, in the coalface, working on these new therapeutic innovations every day. As you survey the cell and gene therapy landscape, what are you particularly excited about? Eric, let's start with you.
Sure. Yeah, thanks. Thanks very much. I thought Sofia did an amazing job, and I always like listening to her, sort of summarize the field. And I thought the key word she used was evolving innovation, and I think that's actually what's happening. I'm actually most excited about the maturation of the gene editing field. Steve and I have lived through both the ups and downs of the whole, whether it's diagnostics or gene therapy world, and I think that initially, as it was with the gene therapy, the viral gene therapy world, people got out of the box very quickly, usually major institutions with lots of money funding it. And what was somewhat missing in those days are sort of the basic science underlying the mechanism of action of how our gene editing tools actually work. And I think we're seeing a little bit of that now. I think the field is, I hate to use the word in a corrective mode, because I do feel optimism, as Sofia outlined. But I do think there are some very serious examples of the science not being quite ready for prime time. Tome was a great example of a company that had hundreds of millions of dollars and basically walked away and fell down in about 12 months. And I think that the companies now that are surviving in the gene editing space, and certainly something that we have really, really based our whole approach on, is understanding how CRISPR works at both the cell and the genomic level. Not only the traditional things like off-target mutagenesis and that kind of activity, but what does it do to the genome, and how does the cell microenvironment, in our case, microenvironment of solid tumors, work? So I think what I'm excited about is we've had our little correction. I hope it's little, I hope it doesn't go on for a lot longer in terms of the financing and having a number of big players fall off the table, frankly, in this world. Bluebird was another one that went down. Relatively recently, a $2 billion valuation company that sold for $30 million to private investors. Just unheard of kind of despair there. But I think when that happens, and what I'm excited about, and having lived through it previously, good science will prevail. And I think investors and analysts will start to recognize kind of a new paradigm, a new shift in how we ought to be doing business. This is not to minimize what those folks did for us. It's very hard to get out there in front and do pioneering work, because you're kind of building the plane while you're flying it, which is a great term for us gene editors. So grateful for all the work that they have done, but I'm really very excited about the fact that I think legitimate and logical progression now of the science will eventually develop into good gene editing products.
Thanks, Eric. And Steve, your thoughts? What are you seeing in the space, and what are you excited about?
Yeah, so what gets me excited really hits me at the heart, having several family members with rare diseases. And I mentioned before, I was in the diagnostic world. Specifically, I was at a company called Athena Diagnostics that acquired by Quest. And that company was really a leader in rare and neurogenetic diseases. And if we think about rare diseases, there are some 10,000 rare diseases. And by definition, that's only 1% of the population for each of those diseases. But you add them up collectively, you're talking about 10% of the population. And so that's profound if we can actually start, chipping away and creating not just disease symptom modifying therapeutics, but actually cures. I had the pleasure of meeting one of the patients who was treated with Casgevy, and hearing, the outcomes that he had, it's just profound. If you think if you can do that for rare diseases, and then of course many of us have been touched by cancer. We think about what we can do with CAR T and how people are applying that technology with now bringing gene editing to bear. It's just, it is the future of medicine, as I think we've heard Peter Marks say. And how do we do this in a way that's thoughtful and we take advantage of both the new innovations that are coming to the field, but do it in a way that's, both efficacious and safe. And so I don't think those two things need to be, counter-opposed to each other. We can accelerate the field. It'll continue to accelerate it, but we can do it in a way that's safe and doesn't create uncertainty and doubt in the minds of again, patients or payers or, the field in general. Right. I think if we do it the right way by working with people like Samantha Margot at NIST, we can start creating this sense of assurance. And so we'll see more investment in the field, more therapies come to market, and we'll embrace the innovation rather than being fearful of it.
Sophia, can you tell us a little bit more about the top challenges when it comes to developing these therapies?
So no doubt that gene and cell therapies are, transformative and bring a lot of opportunities to patients, but there are challenges, and I could stay here for an hour to talk about all the challenges that come along the development of cell and gene therapies. But I think the first thing that it's common knowledge is that these therapies the development of these therapies have associated extremely high costs of development, sometimes exceeding $1 million per treatment. And this, coupled with limited long-term data available at time of approval, makes it difficult for companies to prove and assess their full value and consequently to be reimbursed, right? So in addition to this, obviously, there are challenges in optimizing clinical trial designs, overcoming manufacturing challenges, scalability of production, minimize safety and off-target effects. This is also very important, all the while navigating a regulatory landscape that it's complex, lacks harmonization, is under strict oversight and under ethical scrutiny as well. In terms of the clinical trial side, one thing that I wanted to highlight is that sometimes the need for simpler clinical trials for gene therapies, because these therapies usually target rare or very specific genetic conditions, which make patient recruitment particularly challenging. And if you're still using traditional study designs, these studies will eventually lack the statistical power, the statistical means to gather robust safety data and those responses. So early research and early collaborations with regulatory agencies and accessing real-world data can help to refine and design smarter clinical trial designs to optimize study execution. And in terms of manufacturing and supply chain, obviously, we've mentioned the process of scaling up the production of gene editing therapies. This is a challenging part of the overall scheme. But on top of them as well, they required, for example, cold chain storage, meaning that they have strict temperature controls that are necessary during transportation, storage, and handling to maintain their viability. The other aspect, obviously, they also want to highlight is the safety and off-target effects, because gene editing tools like CRISPR-Cas9 are powerful tools, but also very precise instruments. And achieving this level of precision is very important, because otherwise you will be generating off-target effects. So you need to be very precise to avoid, for example, unpredictable mutations or genomic instability in the long term. So basically, this could be some of the challenges that I feel like I'd like to highlight. But again, these therapies come along with a lot of hurdles that still need to be overcome.
Sure. Thanks, Sophia. Eric, CorriXR is very focused on drug resistance. Why is this an important area of focus in oncology?
Yeah, thanks, Matt. I think we chose to address sort of drug resistance after we spent some time doing something a bit unusual in biotechnology, and that's actually, as an early stage company, spending time with patients and cancer patients. We had the opportunity because we were baked inside an NCI Center of Excellence, so patients were coming in to be treated. And the good news at Christiana Cares Cancer Center, that's actually a community healthcare center, which means that we see folks from all levels of socioeconomic. So we can hear what patients want, and it was really surprising. The idea was they want to just reduce their suffering. Patient suffering has to count for something in our business.
Mm-hmm.
I think not the folks here. We've obviously had long track records of appreciating that, but sometimes technologies are developed for the innovation of the technology. And so we reversed it and said, what exactly do patients want? And almost universally, people believe in radiation and chemotherapy and targeted therapy and some immunotherapies, but the side effects of those are very serious. And so what tends to happen is patients will enter into the chemotherapy trial or immunotherapy. And with immunotherapy, it's interesting, it's actually the preparatory steps that are required. Casgevy is a great drug, but it also takes 3 months of heavy loads of chemotherapy to prepare the patient to take that drug. Now, it's a decade
Mm-hmm.
treatment, so I would imagine that most patients are really, really willing to do that. And I agree with Sophia and you, Matt. It's been a great achievement, and a rising tide lifts all of our boats here in the gene editing world. So we were delighted. But actually, as we looked at the oncology field, and we have wandered into a space that doesn't have a lot of other companies in it, frankly, using gene editing. But we set our goals fairly low in the sense that we wanted to do good proof of concepts. And one way to do that is to reduce the amount of chemotherapy or radiation that a patient has to take. And surprisingly, through 7 or 8 years of work on a gene called NRF2, this is not an unknown gene to pharmaceutical companies, but has traditionally, the product is the transcription factor that gene encodes has traditionally been undruggable, so to speak, because of the way the structure of the protein folds. And yet, it's at the heart of creating a lot of drug resistance. And that resistance goes amazingly through chemo and radio and even immunotherapy. So we had developed a proprietary CRISPR that very effectively disables this, and the tumor by itself shrinks a little bit, but then the tumor, at least in our animal studies, gets wasted away by the subsequent treatment of lower doses of chemotherapy. So we thought drug resistance was a good place to start. We do believe long term that this could be a paradigm for perhaps other ways to treat it. And we also introduced the concept of direct injection. So our focus is on head and neck cancer at the moment and esophageal cancer. I think Sophia alluded to this, that delivery is a very big problem for biomolecules. And our friends at Intellia and Verve are doing great jobs with, targeting rare diseases of the liver, because that's where most of these biomolecules go. It makes perfect sense, except when you have to keep treating them, and even the liver then becomes a bit of a toxic wasteland. So the most effective way to introduce any drug is to directly inject the tumor. Agreed, that limits us to certain kinds of tumors, solid tumors of the oral cavity. But the compassionate note there is that head and neck cancer is the highest cancer with a suicide rate. And so people get disfigured. It's very difficult to breathe. And so we thought we could help. And that comes back to the concept of developing an innovative tool that can reduce patient suffering. So we thought drug resistance was a good place to start. And we've actually discovered a site to target within NRF2 that actually is a bit agnostic to indication. So as delivery improves, we could go after pancreatic cancer or cervical cancer or something. But we have our hands full right now targeting head and neck, oral cavity cancer, and esophageal cancer. And just to end, I think Sophia actually raised a very important point, and this is why we were a little different. We actually spent a great deal of time making sure that our current site does not have off-target effects, and we've been through the nomination and the validation phase as a small research group, and that's a bit unusual. And we'll bring a CCure in to finish that up, looking at translocations. But our approach has been to be completely thorough in our mechanism of action to know what this particular CRISPR drug does, not only at the genomic level, but at the cellular level. So kind of low-hanging fruit to reduce the amount of chemo that a patient has to go through. But if we can get those patients to want this approach, this is the other thing, patients would ask for this. We thought this was a good thing. And then reduce the amount of secondary treatment they have to take. They would actually finish their therapeutic regimen, and hopefully will make them better. So we're sort of approached it in a slightly different way, more of a patient-centric approach than a technology-centric approach.
Thanks, Eric. You hear lots of companies preaching patient centricity and not necessarily so many practicing it. So
Yeah.
so great to hear about your efforts. Steve, continuing on the topic of safety and off-target effects, this is obviously a critical challenge for developers in this space. Can you tell us how your platform is addressing this challenge?
Yeah, for sure. So first off, the field has the opportunity to create cures for the incurable, and CRISPR and base editing and all that has the ability to create very specific targets, but however, they're not always perfect.
Yep.
And in fact, these off targets need to be assessed as Eric was mentioning and being nominated. And failure to do so can have profound effects and lead to really significant results, adverse events, or even death. And what the FDA is now driving towards is an expectation of an unbiased empirical method for measuring these off targets. And when we think about the impact that not addressing these questions can have for the company, these can be profound. We know companies, and some of them were mentioned here, but I won't name them here, were put on FDA hold for the better part of a year just to address their off-target effects, both in the tissues of interest, but also the adjacent cells that are in the body, and whether or not the break profiles would be the same, and whether or not the mutational outcomes of those edits and off targets would be the same. And in fact, that going on a hold caused, in one case, this company to lose over $1 billion of market cap, for which they really haven't recovered. So, answering these questions early on in discovery, we think is super important for the field, where they can assess, and as Eric had mentioned, not just understanding the off targets, but what are the mechanisms of action, the editing kinetics, and the like. Our technology can actually provide this in a scalable and unbiased way. And so we do this in large part by being able to assess all of the endogenous breaks that are naturally occurring, and then using a PCR-free, unbiased way to assess all the breaks that happen after editing. The difference then will be what you induced, hence the name of our core technology, InduceSeq. We can do this in cell, which was going to be novel. We can now do this in the cell of interest that people are working in and actually using their therapeutic guide in those cells of interest. What we've seen a lot of folks do from a development standpoint is use proxy cells because they were easier to work in. And just by virtue of moving into an in vitro environment with a proxy cell, you're now not mimicking what's really going to be your therapeutic outcome or your hopeful therapeutic environment. And so moving into this more unbiased, automated, frankly, scalable way in your cells of interest with your guides of interest, we think will transform and really be disruptive and a significant use to the community.
A great point.
And now capitalization and partnerships I think came up a little bit earlier, and these are difficult tests for any biotech operating in the financial environment, let alone gene editing companies, which may face particularly stratospheric development costs. How have you differentiated yourself in the marketplace and captured the interest of potential investors and partners? And what advice would you give to earlier stage startups for weathering the storm? Steve, you probably have some thoughts on this.
Yeah. So first off, we looked at other companies who are bringing like technologies to market. SecureDX being one of them, which you may have heard about. They basically had an academic protocol that had been a bit cumbersome to use. We've seen it, though, be useful with some approvals in the past, namely the Casgevy approval. And we heard from those folks that in fact they received a requirement for a license. That wasn't taken up front, and we'd rather approach the market in a much more transparent and collaborative fashion. We're not asking for licenses up front. We're asking for an opportunity to work with you and be designed in. One of the things that I think is important for investors today as they think about the appropriate use of cash and capital. And I think these early pioneers that came into the market had to do everything, right? They had to pick their therapeutic area of interest. They had to develop the nuclease. They had to develop all the editing technologies, their own manufacturing, and really verticalize this entire field. And thanks to them, they've done great work. But as we know, when we look at other pharma companies or even other parts of various markets, people tend to go into picking partners, right? And if you look at, for example, big pharma today will use CROs for clinical trials. They'll license technology from smaller biotechs, so on and so forth. But these early pioneers in the CRISPR space had to do it all. And I think we're seeing this retrenching of, to some extent, them down to their lead candidates. But I also think we need to think about how they're going to look at partnerships as a way to conserve capital and get access to the needed technologies, whether it's assays like ours, whether it's going to be delivery systems, whether it's going to be novel nucleases. I think we're going to see partnerships become, a key attribute of this field going forward. Investors are going to want to see that and make sure they're picking the right partners of choice.
Thanks, Steve. Eric, how has CorriXR survived in a difficult financial environment?
Yeah, well, I think it's a great question, and I think Steve really outlined the field and the situation. I can certainly echo that partnerships are always on early stage companies' mind because we've we had the good fortune of being baked into a health system that wanted to really take genetic medicine, CRISPR, TALENs, even zinc finger nucleases, all of which we've worked on for years, and take a solid look at where we could actually be effective. So we did have some opportunities to have a friendly face standing behind us. I didn't actually believe in spinning a company out and raising $20 million, which we could have, and do bricks and mortar. Rather, we have a sort of a relationship with CorriXR that allows them to fund very targeted projects within the research institute. And we tried to stay lean and mean during that period. We've hired, I think, some very good people, a couple of former executives from Spark Therapeutics, and are now chief operating officer and legal counsel. So we have I think we've been very selective, and I think surviving in that way has been good. But we also had 7 years of development of the idea, which was funded by NIH and NSF, and those were the days, right? When you
Yeah,
could automatically
seriously.
apply for those, and these are not SBIRs. These are R01s and R21s. So I was trained as a yeast geneticist, so I was always foundationally baked into DNA repair and recombination systems, and so I knew that if we could harness the power of those cell systems, that would be great. So we had 7 years of really understanding our target and understanding what to do, and we really focused on NRF2 for a long time. But I think in terms of differentiating us, we're from Delaware. We're about 30 miles south from the Philadelphia market, which is quite big, and our friends and colleagues up there. And also, I think we decided that one of the things, and Sophia and Steve both touched on this, that you've got to get the product to the right tissues at the right time. And Steve referenced doing off-target on the real cells, and he's absolutely correct. Doing off-target work on baked-in cell lines is not the wisest idea long term. So we decided to deliver them, a CRISPR and maximal by doing intratumoral injection. And again, as I said, for CorriXR, there's enough targets that that's a common practice, esophageal cancer, oral cavity cancer. But we also realize it's not going to be every possible target. And even though this is a bit of a platform approach, we've been realistic. So I think our own self-checking, our own reflection on what we can be has helped us navigate this definitely financially challenging time. So I think that's important, and my colleagues on the panel had mentioned to think carefully advice is to think carefully about the downstream effects. We've just engaged Danaher as a manufacturer for all of our products, and that took almost a year to work with them and developing how that goes. And as Sophia mentioned, that is a major step forward. And it's a huge education in the scale up of the possibilities and the lack. We had to abandon a couple things that were, we thought were great ideas, but they just simply weren't scalable. So realism, self-reflection, and being willing to stay lean and mean at this time, I think for a company like CorriXR is absolutely at the core of the value.
Mm-hmm. Sophia, what sorts of challenges are your clients in the space struggling with? What are you working on with them?
Okay. Yes, thanks, Matt. So drawing on Clarivate's breadth and depth of data resources, what we see is that companies are reaching out to us at the heart of very early development phases, seeking cutting-edge resources and also strategic collaborations. And, being here today, I've been learning so much as well. But there are some things that we kind of see happening is that the need for differentiation and the need to branch out to other areas, other indications, similar to what we've seen with CAR T cell therapies, slightly moving away from oncology. And you see a lot of clinical development research projects testing its utility in cardiac failure, fibrosis, HIV infections. So the types of customers and projects that come our way usually are related to disease understanding. So we've partnered with a startup gene therapy company to gain deeper understanding of specific disease mechanisms. So we provided this company with systems biology content through a platform called Metabase, which includes detailed information on genes, on molecular targets, on genes, disease pathways, molecular interactions, and metadata. Other projects that we've done as well was to work with an RNA editing technology mid-sized company, and they wanted specifically to identify and prioritize disease-associated gene variants. And Clarivate partnered with this company, and on a very basic level, we basically built a pipeline for them, combining biomarkers intelligence and these systems biology content available within Metabase, and then a lot of statistical analysis to be able to prioritize and identify those disease associated variants. More recently, we are working with a quite interesting project with a precision medicine company that is developing as well RNA targeted therapies and gene editing tools for Duchenne muscular dystrophy. One of the most significant challenges that this company was experiencing was to understand the patient genetics profile. So in the past, they were contracting with multiple laboratories in the US to obtain their genomic data, but this approach was only covering a fraction of the entire patient population. So to address this, we struck a deal with several genomic labs in the US that have genomic data. And we worked with them to standardize this data while integrating it with Clarivate's repository real-world data. And by doing so, we helped the company to better identify the right patients for the right therapies as well, for their right therapy. But in addition, we've also partnered with an external provider to help us de-identify claims data. And by doing so, we were able as well to provide valuable insights into treatment patterns, patient journeys of these patients, even before any genomic testing. So these are some of the kind of partnerships that we have engaged with clients, mainly in very early stage, trying to understand disease, genetic profiling, and with the intent to really apply and differentiate in many other areas of research.
Mm-hmm, mm-hmm. Setting them up for success.
Yeah.
Eric, um, you know, how are you navigating these largely uncharted regulatory waters? How are you in conversation with regulators?
Yes, very much so. And I think I made a, I wish I could claim it, but it wasn't me who first said it. But I think those of us in the gene editing space and maybe in the, detecting off-target mutagenesis space with Steve is we're kind of flying the plane while we're building it. And
Hmm.
it's difficult. And it's not a knock on the FDA and it's not a knock on regulators because this is all really new. The original gene therapy stuff was not this. It was gene addition, although some genes did integrate, and that was in some ways a little easier in the sense of the type of genomic damage that could or could not occur. So we first started by spending about 7 years, frankly, really understanding this tool and our target, published widely on it, really, develop a patent portfolios, and then really took about 7 years to convince ourselves we might have something good here. And we were able to build up enough preliminary data to actually have an interact meeting with the FDA. And ironically, they not only were very encouraging, but they changed our target. They thought that we should focus more on head and neck and esophageal cancer because they liked the idea of absolutely delivering the maximal dose to those specific targets. And so that was helpful. I also think that in this sort of regulatory path, you kind of wanted to discover something that, to use a hockey term, gives you more shots on goal for success. So there are, a great deal of work going on in rare diseases where with gene editing companies, very noble efforts and very, and some quite successful. But in some ways, that's a single shot on goal. If that gene doesn't respond properly, then it can be problematic. Not that they should stop or anything, and we root for them all the time, believe me. But for us, we wanted multiple shots on goal. So we found a multiple opportunity to disable our target gene, which because it's the same formulation and actually even the same sequence, it's a single drug. So I think keeping it simple and directing it makes us navigate the regulatory challenges, which are still significant. There's no doubt about it. But we sort of dumbed it down, if you will, to make it a very simple drug. Again, augmenting standard care, so these patients actually that actually we use more of the drug, but they're at a lower dose, so they're able to maintain their health going forward. So I think those were the ways that we primarily navigated this path, and hopefully it'll be successful. The environment continues to change almost daily, and you got to stay abreast of all of those changes.
Hmm. Keep moving the puck forward.
Right.
Uh,
Right.
Steve, your thoughts? What do you how are you faring in this regulatory environment?
Well, I'll stay on the hockey analogy, and the Wayne Gretzky quote, right? Quote is, go where the puck is going,
Right.
and the puck is going to an increased stringency around, safety and understanding,
Yeah.
the entirety of the workflow from a safe and appropriate way. And to that end, right, luckily, prior to my joining, Broken String had partnered with a group called HESI, the Health Environmental Safety Institute, which is a consortium of many large pharma, including AstraZeneca and Novartis, as well as NIST and FDA. And we were part of a large study that we'll see the fruits of that come out in publication here in bioRxiv shortly, where effectively we took 5 of the most well-characterized guides in the field, and the one for Casgevy being one of them. NIST had prepared those samples, sent it to AZ and Novartis to go do the editing, and then send us those samples in a blinded fashion. What we're able to find is that not only could we reproduce these results, we were able to identify every on target as well as all the off targets in a highly concordant way across 2 different sites. And what that really proved relative to the previous technologies that were out there that I'll call them more homebrew in nature was that we now have a technology that can really become ultimately a standard and a gold standard for this kind of assessment. Additionally, the ability to look at the different on and off target profiles in different tissue types in a simplified and unbiased fashion also turned out to be quite profound. And then lastly, once we initiated this first validation phase, the challenge was what was your sensitivity in your ability to see rare events. And we were able to then take the study into the next phase where we did error-corrected sequencing with a technology called duplex-seq. And we're able to see that we're able to get detection limits about an order of magnitude greater than what you'd be able to see with whole genome sequencing. So again, this effort that Eric had talked about, where he had put a lot of effort in and a lot of time and expense, that's time that could be shortened to either provide valuable treatments to patients or get companies to market sooner so they can go and create and accelerate this field. And so to us, this is compelling, and we know that the FDA does not want to say basically trade off speed for safety. We think we can provide them both. We have been working with many partners in helping them with their IND packages. That's profound. We're hoping that some of these partners here will shortly be able to tell their good stories and of course, under confidentiality, we're not able to do that yet, but we will be shortly. But we are also working with folks like the IGI, and I think it was mentioned, some of the folks in the rare disease field, like what's going on with UPenn. We're going to be doing some stuff with those folks as well to help really set the field for maybe a new umbrella style of approaching or a platform style of approaching, bringing these therapies through to patients in need, whether it be rare disease or cancer. And so super exciting. We're involved with this every day. We're right squarely in the safety area. And again, helping customers navigate their INDs so they get successful outcomes is core to what we do.
Great. I want to take some audience questions as I know we're getting a little short on time. First of all, Eric, I wanted to ask you. You indicated that CorriXR Therapeutics developed in a different way than most gene editing companies. Could you expand on that statement a little bit?
Well, I think that, as I said, we, having been in this field for a long time, was pretty familiar with kind of the history of it and kind of the challenges and the obstacles and the hurdles that need to be crossed, and maybe even some insights to what's important and what's not, even though I question myself every day whether those are right decisions. But I think one thing that, having been baked into this community cancer center, is that it really is about people. And so if you build it, you want them to come. And so how can we really help the patient go first? So instead of sort of setting up a technology-based one, it's ironic, I think Sophia can probably confirm this, but I do believe that all the gene editing trials that are currently underway still use the original strep pyogenes Cas9 that was published in 2012 or something like that. It's amazing. So we believed in that. It's a well-known thing, and we understand its mechanism of action. So I think building the company based on the patient first and coming out with a drug, drug candidate that looks like it can help in many different indications, really positioned us a little differently. And I think, Matt, as you recognize, this is a very common thing, but the tangible evidence, everybody wants to do patient diversity and stuff, but the tangible evidence of that is, at least in the therapeutic development zone, is hard to find, and I think we might have captured that. So I think that's the biggest difference as we differentiate how we go forward. Again, patients need to want to take this drug, and they need to know this isn't going to make them sick. And so I think the opportunities there are greater than just telling them, take this, close your eyes, and have a glass of water. So I think that was positioning how we were thinking about that.
Thanks. I have another question for Steve. CRISPR and other genome editing methods enable the creation of much more complex therapies. What are some of the challenges that this field has to overcome?
Yeah, I think one of the challenges you mentioned is that many people were chasing the same indication in, I think, Eric, you brought up the liver, right? Obviously, there are other tissues of interest, other conditions we need to chase. And so how we think about the ability to provide, as what we know the FDA is going to require, an empirical, unbiased approach that's scalable.
Yep.
This is a real challenge. And I think today, the approach, what we'll say is sort of an empirical, slow approach, is really, we need to move beyond that as a field. We need to move into more of a data-driven science, if you will, turn this into an engineering problem that's scalable. And we're going to get there through the use of tools like AI, through having a standardized approach. Additionally, we need to do the testing of this characterization, as Eric called it, much earlier in the process. Right now, we see a lot of people thinking about, for example, safety at the end, just before they do their IND. If we can, as a community, move to safety by design, so at the same time we're picking our on targets, we're also now determining what the off targets might be. We may sacrifice a little bit of efficacy for having a much better safety profile. If we do that, we're actually to save tons of money across the entire value chain, and again, shorten the time to development. And again, we'll do this by having a much better, more unbiased view of how DNA damage and repair works, which is really at the core of what these CRISPR or base editing systems work with. By the way, I will put in one more plug relative to our technology. We're equally as relevant for base editing and some of these other novel nucleases and some of the legacy ones that Eric had talked about as we are for CRISPR.
Okay.
Again, it's just a function of the way our technology is designed.
Okay. And I think this one's for Sophia. You talked about challenges and method and mentioned the lack of regulatory harmonization worldwide. Can you tell us what strategies are regulators implementing to streamline the approval process and influence the speed at which patients can access treatments?
Certainly. To address that question, I think one thing that it's common practice worldwide and we see across many regulatory agencies is the willingness to work with sponsors as early as possible in the development journey. As it stands, there's not so much like real defined strategies in place. This field is still maturing. We're still monitoring long-term safety and efficacy effects. And as the technology, the therapies reach the markets and mature, regulatory agencies will be able to adapt accordingly into the types of guidelines that they released for human genome editing or cell therapies and other technical aspects. But for example, earlier last year, in January 24, the US FDA has released the human gene therapy products incorporating human genome editing guideline. And here they outlined key recommendations for the design, the manufacturing, monitoring, non-clinical safety assessments, conduct clinical trial designs. So the goal was really to engage and to help sponsors engage with them early in preclinical planning and provide safety monitoring continuously. Likewise, the European Medicines Agency currently offers like what it's called like a task force and provides scientific advice for companies currently operating in this field as well. So they stress the importance of long-term follow-ups for gene editing products to monitor their safety and efficacy as well. But so far, in terms of harmonizations, it seems that some of these recommendations, guidelines being released by authorities are still very much dependable on their own restricted markets. One thing that I want to highlight now that you've asked this question, and Matt, you've referred this as well at the beginning of this panel, is that US and mainland China lead the way in terms of innovation for gene editing therapies. And that the number of patents almost increased 60-fold, right? And something that we've seen is that China is actually progressing towards international harmonization since 2015. And back in 2019, they've created what they call the China Regulatory Science Project, in which they have assigned 9 strategic areas for focus and development in the future, and one of those areas pertain to advanced medicinal products such as gene and cell therapy products. So the registration pathways are now so now what they are doing as well is to update the various mechanisms for regulatory pathway approvals, okay, and introduced recently guidelines in early 2020 to facilitate this process as well. So in essence, I think as we progress in this market, in these areas, we will probably see more and more guidances and recommendations evolving, and hopefully it will become more harmonized, as we see these days with real-world evidence, for example, applicability of real-world evidence in drug submissions, for example, and how different regulatory authorities worldwide are trying and harmonizing over time the requirements for that.
It's nice to see things coming together in a world that is sometimes feels like it's falling apart. Well, unfortunately, on that happy note, that's all we have time for today. If we didn't get to your question, we will try and get back to you via email. I'd like to thank all our panelists today for a stimulating discussion. Keep pushing on those frontiers of medical science, and back to you, George.
Excellent. Thank you, Matt. And thank you once again so much to our esteemed panel of experts. But as Matt mentioned, unfortunately, that is all the time we have available for today. That was a fantastic discussion, and we are incredibly grateful to all of you for sharing your expertise with us today. And as Matt mentioned, as a reminder to our audience, any questions that we didn't have time to get to will be sent to the relevant speaker after the event. And as with all the other talks in the event, this talk will now be available to view whenever you want for the next 30 days. So that just leaves me to say a massive thank you once again to our panelists, Matthew, Sophia, Eric, and Steve, and to everyone in the audience. We'll be back soon with the next talk.
Thank you.
Thank you.
Thank you so much.